First of all, thank you very much to the organizers of this event and for inviting me to join you here. My topic today is quite different from the first two, but I hope to share some of the things we've been doing since I moved to Buffalo 15 years ago. Actually, before coming here, I was in Kansas and I was studying pesticides and in the farms, in corn and antibiotics in swine manure and all that. And coming here, I was wondering, what am I going to do in UB? Because there's no cornfield, there's no swine. Little did I know, I realized there's the Great Lakes and there's a lot of problems that I could really try to study. And also I learned that New York is actually the third largest dairy producer in the US. So I switched from studying swine manure to dairy manure. But it's all about emerging contaminants. And so what I'm going to tell you about is what are emerging contaminants. And for those of you who have never heard of this term, these are actually chemicals that we have in everyday life. And it's called emerging contaminants, not because all of a sudden they just emerge in the environment. It's because of the latest developments in analytical methods. So when I was doing my PhD, we didn't have this analytical tool called LCMS. It stands for liquid chromatography mass spectrometry. It sounds difficult to pronounce. But so when we didn't have that before, we couldn't see any of these antibiotics, pharmaceuticals, personal care products like triclosan in your toothpaste, estrogens that are active ingredients from birth control pills, and also natural hormones, because the tools back then does not allow us to determine these chemicals in water soluble, that are very water soluble in these types of samples. So we could only see pesticides that are applied in the field and so on. PCBs we could see, I did a lot of PCB work. So actually at the beginning I was studying PCBs in the Great Lakes. We would collect fish, analyze PCBs, all that. And then back in 2002, this new tool became widely available and we started using it. And I met some great people from back then, Fredonia was actually here too, a fish biologist from Buff State, Great Lakes Center now. And so together we started looking at fish from Lake Erie. So that's what I'm gonna talk to you about today. So emerging contaminants, one of the things about this emerging contaminant is that they have, actually I could see here, they have adverse effects even at very low levels, meaning nanograms per liter. So to put that in context, PCBs or DDTs, they occur in the environment at sometimes parts per million level. This is at parts per trillion, so at 1,000-fold lower and another 1,000-fold lower, so at very low level. And yet, they can have adverse effects in fish and wildlife. I'm only going to talk about the pharmaceuticals and maybe hormones here, but something that I would like to point out are the engineered nanomaterials. If you have sacks that has that says antimicrobial, they would have silver nanoparticles that could be washed off during and get into the surface water as well from wastewater effluents. The new TVs, Sony, that are really nice would have quantum dots, which are engineered nanomaterials. They have cadmium metal, so in the end, at the end of their lifetime, they might be in the landfill and they might also release some of these engineered nanomaterials, which are also considered as emerging contaminants. So we also study those. So I'll tell you that two things about our study. One is flame retardants that are brominated flame retardants, which back there, actually just prior to 2015, we are still using it, but after December 2015, it has been banned and removed from manufacturing. But so we have now new flame retardants that are maybe more environmentally friendly, but still accumulative. But what we have been doing, studying in the past few years, is the brominated flame retardants, which is here, this chemical. I have to put up a chemical as a chemist. But they're everywhere in the environment. Carpeting, sacks, pictures here, cars. And the thing about them is they're actually not chemically bound, so over time it kind of dissipate in the air and we could inhale it. So I'm pretty sure I could bet my salary that every one of you here will have some PBDEs in their blood. And the thing about these chemicals is that they are bioaccumulative, so they're just like PCBs. They would accumulate in fatty tissues in fish. And this actually, I have put together here some of the typical concentrations in fish that have been published in many years. So there's in 1980, we found something in the literature with 0.3 nanogram per gram weight. And then 2009, it's a little bit increased to 13. But what I'm going to share here is some of our studies. So we collected from the eastern Lake Erie, which I saw from your slide, it has the red stress line. So that's actually one of the reasons why we selected the eastern Lake Erie, because of the pollution that is well known there. And this graph is just a nice representation of a nice summary of what we found. That you can see here, we collected, my collaborators here, we collected from sediments and several types of fish all the way to walleye, and you can see an increasing level of PBDEs as in the food web, okay? And that's very typical of persistent organic pollutants like DDTs and PCBs. So PBDE behave the same way. And while they're very low in concentration compared to PCBs and DDT, at very low levels, they have been shown to have neurobehavioral effects in children. And so because we accumulate them, especially for nursing mothers, they could transfer this to their children through breastfeeding. And there's been a study that is a few years old now, but it's one of the largest study that has been done in the US that followed mothers from prenatal exposure to all the way to childhood, the children up to 7 years, I think. And they have seen effects that are similar to those of the ADHD problem, in the ADHD problem scale. So poor attention, slow fine motor coordination, and impulsive behaviors. So this is one of the reasons why PBDEs have been banned. And yet today, even if they've been banned, we still see them in fish from the Lake Erie. The next thing I'd like to share with you is our more recent study that's been only like last year, actually, and this has not been published still yet. So my collaborators who are here, Mark and Alithia, Mark is in the audience here, Mark Lafsell from the Puff State, they collected some of the fish and water samples, and my students joined their group. And these are the different areas from the Niagara River. We also collected wastewater effluents and before and after treatment, and we analyzed them for some of these pharmaceuticals, and we also dissected the fish, look at their brains, gonad, liver, and muscles, and so on. And these are some of the examples of the fish that we collected. So there's a variety of them, largemouth, trout, and so on. And again, I have to put up the chemicals, but I think a lot of this you will be familiar with the names. So in this circle here are active ingredients of antidepressant drugs. And there's other ones, antibiotics, caffeine from coffee, and antihistamine, and some over-the-counter drugs. So there are many others that we could analyze, but there are some of the ones that we detected. So I think this is the easiest way. I have just 2 more slides to show you that the highest concentration we have seen are the ones that are active ingredients of psychiatric drugs, antidepressant drugs. So this, they go from even all the way to 1,600 nanograms per liter, so it's almost parts per billion. So these highlighted ones are all, and they are detected in almost all of the sampling sites along the Niagara River. We also detected antibiotics and antihistamine. So, and this graph, this is my last graph, I'd like to also point out here, so I kind of differentiated it based on the body parts of the fish. So you could also see that brain and liver are the ones that with the highest concentration. Now, interestingly, these are psychoactive drugs, so they affect the brain, and the fish accumulate them highest at the brain and the liver. So again, these are all kind of new data. We haven't published this, but there are some other groups in the other parts of the US that have observed similar things. And whether or not what the effects on fish are, we haven't looked at that, but I could only imagine that, happy fish with all this antidepressant, they might just go to the predator and not worry about being prey. So that's all. I'd like to thank I got several funding to work on this seed grant, and Water Environment Research Foundation, EPA, USDA, and National Science Foundation. So thank you all.